Yeah, they use a lot of those, especially with the hospitals here. They
Yes,
really like
yes,
to use those.
yes. It was amazing. I mean, this the last 5 years. Okay, there's so many things that happen. Okay, even the genetics. I think yesterday I saw an article that said, and now they're working for the Parkinson
Hmm.
genetic. Okay, they're still working on it for a long time.
Hmm.
But one guy, I think, if you recall, his name is He Jin Kui from China. He is the one that do, okay, the genetic for a baby. And then he got cursed, okay?
Hmm.
The whole world doesn't like it because there's no rule at the time, okay?
True.
He was jailed for 3 years, and
Hmm.
now he came back. He came back, he said, I'm going to follow the rule to be a good guy.
And there was no rule at the time.
Yeah, yeah. So with that in mind, I don't want to take more time from you, Professor Pence. Okay. Now everybody say, okay, at least now here, good morning to Professor Pence. Okay. We need to respect our teacher. All right. Welcome again.
Good to be here. So today I didn't want to start anything new. We'll go over the answers for the practice sheet I sent out last week. I was looking at some of the ones y'all sent over on the WhatsApp app, and they did really well. I caught a couple mistakes, and we'll go over it today. And I got some more practice too. So again, nothing new today, just more reinforcement of the transcription translation stuff. Let's see. Share my screen. So this is the answers to the practice sheet. We'll go over this first, and then I'll pull up my other practice problems. So I did the strand A with this last week. So just the reminder, whenever you see the 5 prime, 3 prime here, that means that the 5 prime is the start of the DNA, and the 3 prime is the termination of the end of the DNA. So with this strand, we would start at this side because when we create the RNA, it would need to be created in a 5 prime to 3 prime direction. So it would be created in this direction. And then this is what the messenger RNA would be. Now the bottom strand is a little easier because since the 5 primes on the opposite end, we would start with this end and then we would transcribe in that direction. So if you transcribe it, these would be the correct RNA nucleotides that you would get. And if you go down to your chart, so you always know you did it right because the genes to start with AUG, and that means you got the correct start codon. So if you see all the answers start with MET, that's because if you go down your codon chart, MET, the green one here, that's always the start codon. So our next one for strand B, UGU. Let's see where we at. UGU right there is cysteine. And then if I was to do the other three, we have glycine, histidine, and then a stop codon. That's what these three would be. And then for strand C, it's very similar to strand A. You would start with this codon, or this triplet actually, and then code in this direction. And then for the strand D, you would start with that codon and code in this direction. So these would be the right RNA nucleotide you would get. And then uh, down here are all the codons. So I did notice that some people missed, oops, let me get rid of that. This guy right here. So the main thing was that that's an I, it's isoleucine, and that up there is leucine. So these are two different codons. And that's one of the mistakes I saw. But other than that, y'all did very well, the ones that sent me the practice. So I'll send the answers later today. That way you can check if you didn't send me. So I'm going to try to combine some of the stuff that we've looked at. So we looked at great Mendel's experiments where he found recessive and dominant genes. We've looked at DNA to RNA to protein. And then we're actually going to look to see how the proteins work. So for this exercise, I've got 5 different mRNA sequences. I've already transcribed them for you. So these are messenger RNA. So we don't have to do the transcription. We just have to do the translation into a protein. And then from there, I have a little chart down here we're going to look at, and we're going to try to match the strands that we have with the physical trait that will be produced. So let me get rid of this guy. So again, AUG is a start codon. You get look for your codon for UUC. Right there it is. Phenylalanine, PHE. GGA. Yeah, right there. GGA is glycine. CCG, proline, and then we have AGE, which would be a stop. I can spell stop. Okay, so there's my amino acid sequence that's going to be creating my proton. Well, I can talk protein. And then what we need to do, I'll make that red, is match that amino acid sequence with the one in the chart. So it's going to be this one here. So red hair. And then if you look at the label, I labeled that gene, I called that big H. So over here, I'm going to put a big H. So what I have here, I didn't explain this. Um, I couldn't actually use human genes because humans genes are extremely long. They would take up the entire page. Okay, so I kind of made up a similar like fictional gene. So let's say that these are for monsters, and we're going to be looking at monster traits. So that one that we just looked at, the messenger RNA sequence for gene H for this monster, it has this amino acid sequence, which means the monster, if it gets that gene, is going to have red hair. So if we do our next one, AUG should look familiar because that's the start codon. And our next codon is AAA. So look on the chart. We have lysine, CCC, getting proline again, UUU, we got phenylalanine, and then another stop. So now I want to take this amino acid sequence and I want to try to match it for my chart. I'll give y'all a second to look at it. Well, lysine proline. It's actually the very bottom. It's way down there. And I named that one Big F. So I go right there and I would put a big F. Again, AUG is my start. CGU, arginine, CG or GCG, Where we at? A blind. Urethra alanine. UAU. Machine. Yeah, so give you a second to try to find it. Then this one's called uh, H with which was called subscript one. This is because in this case we have two recessive genes. There's a small h1 and a small h2. And then if you look, the match is right there. So I put H and then one. Okay, AGU, my chart. Right there, serenine, GAA, glutamic acid, CAC, histidine, and then stop again. We have two options left, and it's that one right there. This is my small f. So UGU, just to double check, make sure we are looking good. GUC. Question. GU. No, wait, I did that backwards. No, I didn't. UGU is this DM on this one. No, it is. I did that. I did double. GUC, valine. CUC, leucine, and stop. And then it does match the last one we have. And that is going to be our other small h. Okay. So now that we know the amino acid sequence, the trait, and the gene, I was wanting to look at some of the stuff we looked at on day one with Gregor Mendel. So we know that the big letters are going to show dominance, and then we know the small letters are going to show recessive. So if you have two, uh, whoops, try this again. So a genotype is kind of like the written down code for somebody's genes. So let's say that we have a monster whose genotype is HH. So that means that this individual got one dominant gene from his mother and he got one dominant gene from his father. So in this case, both of the genes are dominant So there's only one possible outcome here, and the one possible outcome is this trait. So again, that's my dominant H. So that means this monster is going to have red hair. Let's see. Let's say that we have H, H1. So with this monster, he has one dominant gene and then one recessive gene. So with the dominance, the reason why we call it dominant is if you do have this gene over recessive, it hides the recessive trait. So our dominant trait is red, and then with H1, it's going to be yellow hair. So the red hair in this case is a protein that's going to overpower the yellow and not show the yellow. So even though it has a yellow gene, it's not going to be able to see it. So it's going to have red hair. If we would have a monster genotype that's H1, H2, this is very similar because H2 is another recessive type of gene, which means it's going to have a red hair gene and a blue hair gene. But again, in this case, the dominant hair is always going to overpower the other recessive hair. So red overpowers the blue. So this monster is also going to have red hair. So the main thing here is if you see a large letter or an uppercase letter like these here, that trait is always going to be expressed over the recessive trait.
So
Let's look at your
yeah, so
question.
the dominant gene always detect what will be the result. Am I correct, Prof?
Correct. Yes. So with dominance, this is a special type called complete dominance. But yes, if there is one trait that's complete dominance and one trait that's complete recessive, the dominant will always overpower the recessive. There are a
Now,
few. We're going to talk about something else in a second that we kind of change it up a little bit.
yeah, yeah,
This
yeah.
is okay. Let's do Let me make sure everybody understands what I said. This is called complete dominance.
Right. Now, the worst thing if okay, the age, the complete dominant, not always be a good a good one, right?
Correct. Yes. So we have something in humans called it's Huntington's. Is it Huntington's? Yeah, I think it's Huntington's. This is a disease that is dominant in humans, and it doesn't get expressed until adulthood. So by the time that individuals are having children, they're figuring out they have this gene, and if they have it, uh, there's a really good chance they're going to pass it on to the child. But they don't
Oh,
find it. They don't find out on it. They don't find out about it until later on in life.
right, right, right. So the dominant gene will be there forever, or will it change during the time when the time progress from baby to adulthood?
No, you can't change your DNA. So if you get a dominant gene, it's always there.
Oh, hmm.
Mm-hmm.
So we cannot change that through the gene therapy like CRISPR.
So when when a you can check. Okay, so here's the thing. When you are created, right? It's one sperm cell, one egg, one sperm cell, one egg cell that fuse together to make a zygote. From that one zygote, you are going to make trillions of cells. So your body consists of trillions of cells that all have the same DNA. So you can't change all trillion cells in your
Mm,
body.
mm, mm,
You can
mm.
try to pick and choose some cells to change in your body, but the hard thing is trying to get every one of your cells to change because
It's not possible.
it's not possible at the moment. No,
Yeah.
but you know the man that you were just talking about that got arrested. Um, he changed his daughter's zygote, so he changed the very first cell, and from that very first cell, all of her cells were changed.
Oh, wow.
Yeah, that's what I'm taking from it. I haven't really looked up Um,
Yeah.
yeah, she can go
Yeah.
to the bathroom.
Is that ongoing research still?
Yes. So, uh, CRISPR, there are some CRISPR programs or companies that are sending like kits out to individual people,
Mm,
and individual people can alter their own DNA. But
right.
again, it's not a lot because they can't alter their entire body. They only can alter some cells in their body.
Mm. And it's very costly, I believe. The
It is
insurance will
very
not
costly.
pay.
Yes. Yes.
Okay. Yeah.
The
So
thing is,
guys, so this is the implication for our student. The opportunity is wide, not is, but are wide open for anybody that's taking this class and outside the class, okay, about the genetic. This is the future, guys. All right. You are very lucky, okay, prof fans, okay, at 8 o'clock in the morning, okay, share his valuable time, okay, for you guys. So learn very seriously and ask question if you do not know. Okay, go ahead. Uh,
okay,
well,
so let's keep going with this genotype and look at the other ones. So let's say that we get individuals that are H1, H1.
mm, mm,
Okay, so in this case, this monster genotype is recessive for the first allele, or recessive for the first gene and recessive for this also the same gene. So if you look at that gene, right there's our H1. That means this trait belongs with yellow hair. So it's got both copies of the recessive allele. It doesn't have a copy of the dominant, which means the recessive allele is going to be allowed to shine through. So this monster is going to have yellow hair. We do the other one, H2, H2. Similar process here. The H2 is recessive. So it's got both copies of the same recessive allele that is going to make blue hair shine through. So since they didn't get the dominant allele, the red hair is not being allowed to overpower. So notice I didn't do H1, H2 because that's something else. That's something special. I get that bugs me. I gotta fix it. Okay. So the next one that I want to do is something called incomplete dominance. So I can type. So with this one, we have a genotype of H1 and H2. Okay, so since these are both the same type, they're both recessive, but they are coding for two different kind of hair colors. They're both going to be made. So with incomplete dominance, they actually kind of mix together and they make a new trait. So with our H2, we're going to have yellow hair, and then with our H2 is blue hair and H1 is yellow hair. So they're both going to be allowed to be produced and created. So when they both proteins are created, they kind of mesh together. So in this case, think of blue and or blue and yellow making green. So this monster would have green hair. This is again a special thing that most genes don't do. Um, let's see, we do have incomplete dominance in humans. There's a disease called sickle cell anemia. You might have heard of it. So with sickle cell anemia, the our blood cells form a very kind of crescent shaped moon shape, and that's not very good at carrying oxygen. But if you have an individual that's got the recessive and dominant allele for sickle cell anemia, they make an oblong, like oval shaped cell, which is perfectly fine. And the reason why we keep the sickle cell anemia trait around in our population is because of malaria. So malaria is um, it can affect a sickle cell. So individuals that are heterozygous, so like up here with this one, H1, H2, they actually are immune to malaria.
Mm.
Okay, so let's look at the fingers. We go back up here to complete dominance. So let's say I have a monster genotype type. Let's see if I can figure this out. So if I have a monster genotype, big F, and then right there's my big F. Is it going to have 4 fingers or 5 fingers? What do you think? You can type it in chat. You can say it.
Alfonso?
So what?
Alfonso? Give them a time, question.
Mm-hmm.
Oh, Michelle. Anyone?
Yes, sir. Four finger.
Good. So look, it is a big F, right? So big F is dominant in this case, meaning that it will always have that trait. So you are correct. 4 fingers. What if we change it to a big F and a small F? Does that change anything?
Four finger.
You're right. So
Wow, Alfonso
even
is really hot, huh? He is full grader.
what about small F, small F? Go back to my chart.
Five finger.
5 fingers.
Wow, really hot. Very impressive. This is the future scientist here, Professor Pence.
You're doing great. So this one wouldn't have an incomplete dominance trait because this one's just all complete dominance. So while I have this up here, let's talk about mutations. This would be good. This would be a good kind of breakthrough for mutations. Let's copy this first one. Okay. So the way mutations work, there's different reasons for this. You can have a spontaneous mutation, meaning that your cells just do it by mistake, or you can have something called mutagens, which are factors from the environment, like cigarette smoke's a big one. Um, a lot of chemicals, hazardous chemicals, can be mutagens. So what are these things doing? They're going into your DNA, and you see these nucleotides that are working, like that's the working correct nucleotide sequence. They're changing these nucleotides up. So we're going to keep the first of the top one, the original, and I'm going to mutate my second one. So in mutation, let's get rid of this codon and let's put in a new codon. Let's put in U, I don't know, A, G. Nope, it stopped. Let's not do that one. Let's not do U. A, A, C. We'll do AAC. So in this case, the first two didn't change. So I'm going to keep them the same for my amino acid sequence. But now my third codon is different. So if I go to my chart, it's AAC. Or is it ACC? Which one would I put? Say AAC. That is, what was that one? ASN. And the third one didn't change. And then the last one didn't change. So notice that now this protein sequence is different because this third codon is different. So this gene wouldn't be H anymore. It would be a new gene. We would call it, I don't know, we'll say um, H2, H big 2. So there are many factors that could affect this. This gene that's been created, it could be something new. It could make a new, so these are hair, right? Yeah, I did that for hair. So the original H protein sequence was this one here, and it coded for red hair. So now that this one is different with the ASN in the middle, it's going to code for something different. So we'll say it codes for orange hair. Where the original coded for red hair. That's one possibility. Another possibility is it could not work at all. This is what most of our diseases are. So you have a working gene, and then when it mutates, it could create a non-working protein, which is equal to a disease. And then with CRISPR, what we've been trying to do with CRISPR is we know that the non-working protein sequence looks very similar to a working protein sequence. So if we can go in there and change the mutated gene back to the original, that's kind of fixing the disease. And that's hard to do though, because you have to figure out a way to do that for every one of your cells. If you hit the stem cell though, that's the kind of the go-to or the gold cell if you can hit the stem cell. Do another one. So this one I want to do a little different. I want to get rid of this codon here. See, it's very close to this one. It's UUC, this one's UAG. Let's change this one to UAG. So what that means is these first two codons are red, and the only things that get created are the start codon and the stop codon. And then when it sees stop codon, it stops. So none of this gets red. So all this, I'll do orange. All that in the orange, it just gets left out. It doesn't get red. So this is going to be a very bad trait. This is a non-working trait. And the difference between this one and the other one is it was still the same length. It just changed one codon. This one is no longer the same length. It's completely shorter, which means it will not work at all. It's just one start and one stop codon. Are we good? Any questions? So y'all are, I mean, from what I'm seeing with the answers I'm getting through the app plus y'all's responses, yeah, I mean, y'all seem to be kind of grasping this. What do you think, Harry? You think they're grasping it? Still here?
Anyone can answer that question?
You think we're ready to move on to something new next week? So I'll send this activity in the chat. That way y'all can work on it again by yourselves. I'll leave. So I'll copy this over to my um what do you call it? Assignments, but I'll get rid of these that way you can figure it out yourself. And then next week, you want to move on to mitosis? Talk about how cells copy and divide.
Mm.
We'll talk about stem cells too. So if again with the CRISPR and trying to fix these, it's hard to fix cells that have been created, but there are a few cells in our body called stem cells. And with stem cells, they are not determined what they want to become. They actually can change. So if you can find a stem cell and get that DNA into a stem cell, it sticks around a whole lot longer than any other cell in the body because think about your skin cells or your hair cells, they flake off and you lose those. So if you were to uh, manipulate those with CRISPR, they'll eventually be lost. So you have to keep doing it over and over again. But if you can hit a stem cells stay in the body for much longer. And if that stem cell's changed, then it's going to change every cell that it produces. It keeps the change. So most of the people that are getting the CRISPR in the mail, they're having to let's reinfect themselves with CRISPR over and over again because the changes only last for a couple months. We'll look
So,
at why next week.
yeah. So which one is more efficient, STEM or CRISPR?
What do you mean by STEM? Science
Yeah.
and technology, English and math.
Yes.
Oh, you're talking about stem cells. My bad. Stem cells. So stem cells.
It's longer,
If
right?
yeah, if you want to infect, if you want a change to last long time, or for it to kind of have a bigger hold, you want to infect or you want to treat stem cells
Mm.
because if you can treat a stem cell, it's going to stay. It's going to not it keep stem cells. They're constantly going through mitosis. So we'll look at mitosis next week. And if they're constantly going through mitosis, they're always copying what you change.
Mm.
So
Mm-hmm.
they're hitting that copy button over and over again. And if you throw a new segment in there, they're copying that new segment too. But like if your skin, your hair, those cells you lose. So if you were to treat your skin, your hair, in a couple of months, they'll be gone because you grow
Mm-hmm.
new skin, you grow new hair. Make sense?
Mm-hmm. 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 Okay. Yeah. Yeah. So next week there will be start with the stem cell
Yeah,
discussion.
we'll start with stem cells and mitosis, and then we'll try to tie it in with this here.
Okay. And today this topic is complete, right? So,
Yes, I mean I'm the what I'm grasping is they're kind of getting this with what they've sent me with their answers and then their
honey, honey, Louis and they work.
mm-hmm.
So they basically grabs the idea, and I was surprised.
Me too.
Alfonso kind of clicked it like that. 4, 5, 4. What about others? Okay, we would like to check that. Okay,
I have
guys.
students in my biology class right now that are struggling with this because
Yeah.
this is my last unit was DNA with them and they were which they're not trying. These guys are trying.
Mm-hmm. Mm-hmm. Yeah, yeah, yeah. So guys, you do not want to miss this topic. Okay, you need to understood very solidly, solid understanding about this that will bring you to the next thing. Okay. Now, to be honest, okay, Professor Pence, about this material, okay, do you think that the high school kid in the US have learned this material, this intensive, this deep?
No, because my students in college that are coming from high school, they don't know it. No.
Oh my goodness. You see, guys. So were you surprised to see this fourth grader like Alfonso? Because he is the one that say bam, 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 bam. I suspect that the other, other participant also understood.
Mm-hmm.
Okay. And Honey. and Louis. Okay, remember that what Professor Pan said. Professor Pan is teaching at the college level. Okay, the high school kid that coming take his class, not even understand and heard about this. So you guys is how many years? Okay, if you're 4 years, you staff. Okay, if you are fourth grade, let's say you are sixth grade. Okay, you have middle school and high school, that's 6 years. Okay, so you guys are 6 years ahead, okay, of your peers in the US, not mentioning your peers in Indonesia. Okay, I'm telling you, this is serious. Yeah, Professor Pence would like to see a kid, okay, like you in his class.
I would, yes. I really would.
So why is that? Because it's easier for Professor Pence to, to build his class because you guys coming with something in your head. Okay, compare with somebody that do not know nothing. It need to come back. Okay. So we are hoping, guys, and we are working so hard now, okay, to write a proposal for funding, okay, for any company, okay, any nonprofit organization in the US, okay. So this will help humanity. All right. It doesn't matter where you're coming from. Once you know and find, okay, a certain drug, a certain technology to do this, the whole world will be beneficial, including, but not limited to the US. Okay. So
Yeah, Harry, if you have them send those assignments to you and you save them and show these uh people you want to help fund, I'm pretty sure that they'll be pretty shocked that fourth graders are doing this
yeah,
type of because
yeah.
I was
And
shocked.
and this is the big thing. You are taking Professor Penn's class. What does it mean? Okay, Professor Pence know who you are, like Alfonso. Let's take an example. If Alfonso et al. Say, let's suppose this, right? A dream. Alfonso like to finish his study in the US for the high school. All right, for the high school. Now, would Professor Pence make a recommendation for him to come to the US to finish his high school with all of this? Would you?
Yes. Yeah.
Definitely.
Yeah.
Because you are ahead 6 years, even, okay, when you attended high school in the US. At the same time, you can dual enroll at the university, okay? So we are here, okay, Professor Pence and I and everybody else to help you to come over here, because the world need you. The world needs somebody who can help the sickness. We have all kind of problem in this world, okay? And you heard that from what Professor Penn said, okay? He will support anyone, okay? Anyone to come, okay? To have open better opportunity. And I did not want to say this, okay? But in reality, Professor Penn, okay, they in Indonesia, in a small place, not in the capital, okay, not in a big private university, private high school, they don't have lab, no lab at all to
Mm-hmm.
learn biology and chemistry. How can you imagine how far they are behind? Okay, all right. So this is the only way we can do this, all right, for you to fill the shoes. But the lab, okay, the lab, we need to collaborate with the high school that really want to do this. But most of the private high school, they do not want to share their equipment or the facility to
Mm-hmm.
others. Of course, this is competition, right? It's very sad. So what does it mean? Once we have that lab, when we have that lab, okay, and when we have that funding, I'm not sure, okay, Professor Pence, can we say have the online lab? Is that possible? It
Um,
it has been existed. I think some of the online lab that what I
there are
somehow I
some.
heard.
We have some. We call them simulation labs. Yes. They do cost money. That's the bad thing.
Yeah,
Yeah. Um, so with our online classes, our labs either are simulation labs where you do it on the computer, or there are labs where you go to the grocery store and buy household products and then come home and do it, and you just show us you've done it. That's what kind of our labs are for online.
yeah.
Hmm.
So this is what my plan. I've been dreaming to write this proposal, okay, to the and Melinda Foundation, Illumina, okay,
Hmm.
and all of that. And if you recall, Jeff Bezos X, okay, give 340 million recently, okay, to all this nonprofit. I missed that deadline. I did not
Hmm.
know. I only read that the news. Okay, next year we are going to compete, and we are hoping, okay, Professor Pence will be co-author, okay, PI on this grant. Okay, this is a
Okay.
big dream. All right, now that is the reason why we make all this in record recording. So when we applied for the grant, okay, money for the Jeff Bezos X, okay, I think it's something like growing yield. Okay, yield. What it, I think the name of the organization, and also Mark Zuckerberg. Okay, Facebook, the Chan Foundation. All right, and there are a lot of them still. All right, they don't have any idea what we are doing right now, and they will be amazed. This fourth grader. All right,
Hmm.
like Alfonso, and I know that some of them. Okay, will be able to understand this. So what do you mean? Age and grade doesn't matter, but opportunity matters. When you give them opportunity, they will learn as well as others. Okay. I'm so pleased to see this. Okay. And we need more collaboration. And our dream is to make this, a virtual lab. And hopefully we get the funding. And we hope, most of all, that Professor Pence will help not just this semester.
Hmm. I'll keep helping.
Yeah, this is exciting. This is exciting. And of course, all right, like Michelle Tang, that has been admitted to Colorado University. Okay. She can say, look, I have this background. Okay, please give me more scholarship money. Okay. So we are hoping. Okay. So I better stop over there, guys. All right. You get a world-class opportunity. So do not miss it. Do not miss it. Be serious. With that in mind, I'll pass it back to Professor Pence. Since we are about 10 minutes more, or you have any question or anything like that, please
Yeah,
ask.
everything for me because that was it for today. Because I really thought this would take longer, but
Yeah,
y'all seem to get it really
yeah,
quickly.
yeah.
I was
All
shocked.
right. Anyone? You have, okay. How many of you grasp the idea? Okay, put your hand. Okay, like, the five. Okay, Noel, do you know? Okay, Alfonso, of course. Yeah, we we saw him, right? What about others? Okay, Michelle. All right, two of them. Okay, Luis, how about you? Do you know all the concept because you also did the work? How about
Hmm.
Honey? Okay. Okay, Noel, that's it. Good. Okay. Wow, most of them, honey. I'm so glad to see this. You see that?
Hmm. So I'm going to
Okay.
send this exact same thing we did today, but I'm gonna give you a couple of days because I don't want you to fill it out right now. I want you to uh kind of forget about the answers and then come back to it, and then I'll send it. I'll probably send it. What is that? Saturday? I'll probably send it Monday morning. Okay. Expect it Monday morning, and then you can send me the answers if you fill it out, like with this other activity we did, and I can make more. I'll make more for next week too. just keep you practicing. Because I was shocked. I mean, I have students that are 20-year-old and plus, and they didn't do very well in their assignments.
Mm.
And y'all outshined them by a
Yeah.
mile.
Yeah. Were you impressed? Were
I
you
was
impressed?
impressed. Yes, I was
Yes,
impressed.
yes, yes. So this is the future scientists that we would like them to grow. We give them opportunity with whatever mean that I have. You know, I have very limited. I'm not a millionaire or billionaire, not even close.
Me either.
I'm so glad that, uh, Professor Pence have the similar idea. We are going to give the opportunity and, okay, we are, once you be a doctor or somebody, okay, you can come back, okay, and give back to this and, make the good one, make the new one, okay, scientists. We grow this. This is like the humanity, okay, humanity project, okay, human investment, and we also call it human, okay, progress, okay. So I do not want to take more of your time, Professor Pence. You have
You're
so
good.
many.
So just so you know, Monday when I get to work, I'm rubbing this 4-year-old or fourth grader success in my colleagues' faces because we have students that aren't doing any of this. Y'all grasp it in 1 week. I'm very proud.
Yeah, amazing. Now,
Hmm.
would it be possible though? Would it be possible for them? I know it is kind of for them to to attend this virtual. Because it is like Saturday at eight
Hmm.
o'clock in the morning. Okay, but if some of them, all right, some of them would like to learn more, they are more than welcome.
On
It
my
doesn't matter
students.
who they are.
Hmm.
Okay. Yeah, yeah. So we are going to expand this opportunity to everybody. Okay.
All right, guys. Y'all have a good weekend. Or
Okay. Yeah, it is weekend here now.
yeah, I had to think. What is it today?
Thank you so much, everybody. Thank you so much, Professor Pan. Can you
Welcome.
show some gratitude to Professor Pan? Say thank you. Come on, honey. Open your. You can say something.
Thank you, Prof. Kenny.
Y'all welcome. I'll see y'all next week.
Have a nice weekend.
Y'all too.
Okay.
Bye.
Thank
Bye.
you.
Bye.